Opening speech from Dean of Faculty of Teacher Training and Education. We would like to invite Dr. Mardiana, MSc, to deliver a speech.
Okay, thank you very much for Ms. Sania Aurora, the Master of Ceremony in this activity. The Honorable Associate Professor Dr. Muhammad Akhil Aryan Wong from Unity Pendidikan Sultan Idris, Malaysia. Thank you very much for joining this activity. The Honorable Dr. Bowo Sugiarto, the head of Biology Education Master Program. Ladies and gentlemen, welcome in Universitas 11 Maret, especially in the Faculty of Teacher Training of Education. And I also would like to say thank you very much for joining this guest lecture with the topic of Bioprospecting Microbial Enzyme for Industry. We also thank you very much for the Biology Education Master Program for hosting and organizing this activity. I hope the activity will be benefit from all of participants, especially the student from Biology Education Master Program and the lecture. I also say thank you very much for Professor Dr. Muhammad Akhil Aryan Wong to share your knowledge, experience, and I hope in the next time we also have collaboration between Universitas 11 Maret and Universitas Pendidikan Sultan Idris, Malaysia, especially in the research collaboration, publication collaboration, student exchange, and so on. And we also thank to all participants for joining this activity, and we hope you can enjoy in the guest lecture, virtual guest lecture, and we hope also you have get the benefit of the lecture. Based on the request of the committee, by saying Bismillahirrahmanirrahim, the virtual guest lecture is held by Biology Education Master Program today, Tuesday, 31 March 2022 is officially open. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Mardiana, MSc, for your delightful speech. All the participants are kindly requested to open the camera to take a picture together. Thank you. The next program is keynote speech from our keynote speaker, Associate Professor Dr. Muhammad Akhil Aryan Wong from Department of Biology, Faculty of Science and Mathematics, University of Pendidikan Sultan Idris, Malaysia. Ladies and gentlemen, let's welcome our keynote speaker, and we would like to invite Associate Professor Dr. Muhammad Akhil Aryan Wong and Dr. Umi Fatmawati, SPD MSc, as a moderator to deliver the speech.
Okay. Yeah, thank you very much from the Master Ceremony, Mbak Sania. Assalamualaikum warahmatullahi wabarakatuh. Good morning, the Honorable our Dean in Teacher Training Faculty, Bapak Dr. Mardiana MSI, and then the Honorable our Head of Department in Biology Education, Magister of Biology Education Study Program, Bapak Dr. Bowo Sugiarto MPD, and then also the Honorable of our guest lecture today, Bapak Profesor Madya Dr. Muhammad Akil Aryan Wong from Universitas Pendidikan Sultan Idris Malaysia. Okay, let me introduce myself. My name is Umi Fatmawati. I'm the moderator of this session. Today, we will take a look at the interesting topic about enzyme and from microbe. And also their bioprospecting in manufacture to support human health, human need. We know that the first observed commercial enzyme not come from microbe, but come from animal stomach called renin. I know that most of the students know this renin. The enzyme were used as cheese making production. But right now, this enzyme can be produced in large scale by microorganisms through genetic engineering. So the cheese production also growing rapidly. So we cannot imagine how many cows that should be killed only to extract this enzyme. But luckily, from the genetic engineering method, we can have a large-scale production of this enzyme. Okay, talking about enzyme, it will be more interesting if we listen from the expert. And today we will have a lecture session with Professor Madya Dr. Muhammad Akil Aryan Wong, the enzyme scientist from Universitas Pendidikan Sultan Idris Malaysia. But I would like to read the curriculum vitae from our lecture. Okay, his complete name is Dr. Muhammad Akil Aryan Wong, or maybe the Chinese name, Mr. Wong Cheva. Okay, and then maybe we can, in the nickname, we can call Dr. Akil. Yeah. Okay, Dr. Akil graduated for the doctoral degree. They graduated from University Putra Malaysia with a major in microbial biotechnology, and he also graduated in Bachelor Science from University Putra Malaysia. And Dr. Akil began his career in UPSI from 2008 until now, and he is an associate professor or researcher in Biology Department, Faculty of Science and Mathematics, Universitas Pendidikan Sultan Idris Malaysia. And in 2020, he also became a visiting professor in Universitas Negeri Yogyakarta, Indonesia. And then now he also joined as a professional technologist in Malaysia Board of Technology from 2020 until now. And then his affiliation in Biology Department, Faculty of Science and Mathematics, University Pendidikan Sultan Idris, Perak, Malaysia. And for everyone who will contact Dr. Akil or will discuss something, you can directly contact by email in chifa@fsmt.upsi.edu.my. And then, okay, the research interests from Dr. Akil are microbiology, and then plasmid biology, protein chemistry, and structural biology. Moreover, yeah, in this slide only 5 publication, but I know that he sent me many list of the publication, and most of them were published on the reputable international journal. And the latest publication, and his latest publication is about the estimation interleukin 4 and interleukin 6 level in sera from patient with type 2 diabetes mellitus. And this article was published in Indian Journal of Forensic Medicine and Toxicology. Okay, I'm sure that all of the manuscript is very reputable, and maybe they also have a high citation in his manuscript. And then he also read some books and also book chapter, and he holds some project research in enzyme and protein engineering and bioactive compound. And the last but not least, he also get a lot of awards from national and international event, particularly in scientific invention. And the latest awards got from University Pendidikan Sultan Idris about the development of novel purification strategy from bacterial Escherichia coli and Pseudomonas genetic tools. Yeah, he got gold and special awards in applied science category. Okay, yeah, it will be more interesting if we read the Dr. Akil achievement in detail about the Dr. Akil achievement, but unfortunately we have limited time, so we have to directly go to the main events. Before we start, I would like to say greeting to Dr. Akil. Selamat pagi, Assalamualaikum dokter. Okay.
Pagi, waalaikumsalam, Dr. Umi.
Yeah, thank you very much, doctor. Okay, we hope you always in a good health. Okay, doctor. Before we begin the lecture, okay, let me say that the given time till 12:30, but you can maybe share your presentation about one half and 2 hours, and then the rest of the time we will have a discussion session. Okay, and now I want to invite Dr. Akil to deliver his lecture. Okay, doctor, you can start this lecture. Time is yours.
Okay, Bismillahirrahmanirrahim. Thank you so much, Dr. Umi, Dr. Umi, for a very kind introduction of me. Okay, so first of all, I would like to greet and say thank you and to extend my appreciation, first of all, to your very honorable, the Dean of the Faculty, Bapak Dr. Professor Dr. Armadia. Thank you so much. And also next is also your honorable Bapak Dr. Bowo Sugiharto, the head of department of the faculty. Thank you so much. Nice to meet both of you. And also, yeah, sorry. And also to the master of ceremony, I believe is Miss Shania. Thank you so much. And the next person is also the person that I contact all the time. But and again, I would like to extend my apology to her, which is your honorable Ibu Dr. Umi. All right. For sometimes, yeah, due to some of the schedule that I have a very delayed reply for that. But alhamdulillah, we are destined. Alright, kita ditakdirkan untuk bertemu pada hari ini dengan kalian semua. Okay. Next is also to the members of the faculty in Universitas 11 Maret. Alright, be it the administrator and also to all the dosen, to the lecturers of the department and also to the faculty. All right, and then to the students, all right, of Universitas 11 Maret, I was made understood that there are postgraduate students as well as undergraduate students who are actually in this session for today. And yeah, I think the numbers is increasing. So it is around 92 right now. So I hope the numbers will increase as well. Okay, hopefully, inshallah. All right, and last but not least, I would like to extend my welcome also to my friends, my fellow friends in Malaysia. So I think some of us, I extend some of the invitation to my students as well as to the staff. Okay, all right. And then the next thing is that I would like to say thank you to Ibu Dr. Umi for a very, very kind introduction of me. All right, okay. And also I've sent in a lengthy CV for you, so and you need to read all of that and we will need to summarize it. I'm so sorry for that. But actually, I really feel grateful for the kind introduction. Okay, and also to the Honorable Professor Dekan, Dr. Madia, and for the officiating this program. Okay, it's a great pleasure for me to come for today and join all of you, and to also to appreciate the kind invitation from the university to give this keynote lecture to all of you. It's not a lecture, basically. I would like to say it is kind of like a sharing session with all the docent and also with all the students. All right, so I would like to make this session as a two-way communication. Please interject me at any time if you have any questions. All right, so if you want to wait it until the end of it, is okay as well. Okay. All right. Okay. And Dr. Umi also made me understood that all the students already have a prior knowledge, at least a basic knowledge on microbiology, biochemistry, enzymology, and things like that. So I think things can be explained in further detail. All right, since all of you will have already have the basic knowledge on that. Okay. So without further ado, so please allow me to share my screen. Polanya, I would like to share my screen. Okay. All right. Okay. So may I know if all of you are looking at my screen now? Is it?
Yes, I already
Okay.
see.
All right. So thank you so much for the response, Dr. Umi. Okay. So yeah, all of you may feel very surprised. The screen that you're looking right now is Enzyme Quiz. Okay. So please don't be afraid. So this is just somehow just to refresh our mind, all right, on what is enzyme, on what are enzyme, on what are proteins and things like that. And there are relationships between protein and also enzyme. Okay, so before we start this quiz, so let me share my slide dulu. All right, so yeah, please allow me to use both languages, which are the English and also the Malay, to convey this sharing session. Okay, so if some of you may not understand what I'm saying, so you can just simply raise up your hand, or you can just interject me anytime, so that I can actually repeat myself. Okay, and make it clearer, inshallah. All right. So the task that was given to me by the department is actually to deliver a sharing session entitled Bioprospecting of a Microbial Enzyme for Industry. Okay, so my name is Muhammad Akil Aryan Wong. So just now, some of you may feel a little bit confused when Dr. Umi introduced me as Wong Shifa. Yes, that is my original name because I'm a mu'alaf. Alhamdulillah, I reverted to Islam in 2020, if I'm not mistaken, 2000 end of 2019, sorry. All right, before the pandemic hit us. All right, so on the 27th of December 2019, Alhamdulillah, I reverted to Islam, and right now I feel Alhamdulillah, I'm going through a more peaceful life. All right, and yeah, of course, inshallah, with the guidance of Allah, then we can, have a better life. All right, now and the life after. Okay, selepas kiamat. All right, so I'm an associate professor and also the researcher affiliated to Department of Biology, Faculty of Science and Mathematics, University Pendidikan Sultan Idris in Malaysia. So I think we have with UPSI and also with Universitas 11 Maret, we do have MOU or MOA, I believe, right? That we can have further, like what Professor Dekan Mardiana mentioned to us, that we can have further tasks or work that we can have two-way, all right, in which we can send in our expert, transfer of knowledge, technology, experts, students, all right. So all that, hopefully we can do that, all right, in the near future. Hopefully in a very, very near time now, all right. Okay, so when we have this title, and that is called bioprospecting. So let us look at the prospect itself. The word prospect means that all possible way or alternative that is actually meant for something. All right. So in this case, of course, the subject is the microbial enzyme, and the target of this subject is towards the industry. Okay. So bio. So of course, this is towards dedicating towards the bio aspect, the biological application, all right, of this microbial enzyme for industry. Okay. Ibu Dr. Umi mentioned that the very, very early stage of enzymes or protein, okay, started from animal, which is very, very true, all right, that we call as renin, okay, originated from the stomach, all right, and it used in a cheese production. Okay, but when we talk about animal and things like that, it has certain drawback, a lot of drawbacks actually. So that is why we look into different alternative, different way, different bioresources, because Allah have actually granted us with this biodiversity, so that we as the hamba Allah are actually exploring into all this biodiversity and make good use of it. All right. So one of the biodiversity from this enzyme bioresources is of course from the microorganism. But actually, I will talk about this in a later slide. All right, that we will talk about what are the kind of sources that we can obtain the proteins and also the enzyme for the use of human life and also in the industry. Okay. So maybe I can show you. Maybe the view is not very clear because some of the panel are actually covering the images. Okay, so if you look at this, sorry, I'm not sure whether I can close this. Oh yeah, I can close. All right, so if you look into this slide alone, all right, other than my name, so you can just put aside it. All right, the image on your right, okay, is actually one of the 3D image. All right, the 3-dimensional image, the actual structure image, all right, that I obtained when I did my postgraduate study, a postdoctoral study, all right, my postdoc in Kyoto University in Japan, all right. So this is one of the protein that is called beta-4 subunit protein. So this protein is actually derived from ion channel in human, all right, that we call as voltage-dependent calcium ion channel. And this ion channel actually govern the neurotransmitter release of the neurotransmitter, the chemical, all right, in to send in messages, all right, from our synapse to the brain, to the central nervous system, all right, and we have the response of it. So this image, all right, basically it is done through the technology, all right, that is involved in biology that we call is biotechnology, all right. We express the enzyme, we purify the enzyme, and also we crystallize it, all right. So this is basically the crystal structure, all right. So what you can see over here is actually the crystal structure of the beta-4 subunit.
Yeah,
Sorry?
excuse me, doctor.
Yeah.
The slides is not moved, so we only see the first slide. So maybe.
Yeah, this is in the first slide. So because the image that you can see over here on your right.
In the first slide. Oh, okay.
Yeah, still in the first slide.
Yeah,
I'm so sorry.
Yeah.
We are still in the first slide. Okay, I'm so sorry.
Yeah, thank you, doctor. Yeah.
But just to confirm whether all of you looking at the same this blue color ribbon structure, in blue green structure, and over here there's a red structure over here. Is that true? Are you looking at this image?
Yeah.
Okay, all right. Thank you so much.
Yeah.
Yes. So this is a 3D structure of it. All right. The beta 4 subunit. Okay. That I actually crystallized the protein. All right. And eventually I shoot it with the X-ray diffraction. All right. So this is the orang kata terjemahan ataupun or the representation of what we have in the crystal structure. I will let you know what is crystal structure because some of us may not have the idea whether the protein can be crystallized or not, because most of us understood that protein, I mean just salt, for example, garam, okay, can be crystallized, right? Or even some of the gemstone can be crystallized, batu permata yang boleh crystallize. But actually, to a surprise, actually, protein can be crystallized as well. And protein crystallization is one of the very, very advanced method for us to identify the structure of the protein and eventually look into the structure of the protein and study more. Okay, because we can do some mutation or to change the structure so that to fit in to our characteristic that we want. All right. Okay. All right. So now we are moving to the next slide. Okay. All right. So yeah, I think before this, all the students in Universitas 11 Maret, all right, already been through some of the lecture from Malaysian lecturer. Is that true? Because I understood that one of the lecturer in biology department as well, Dr. Haniza, Associate Professor Dr. Haniza also gave a keynote lecture to the students in this department. Correct me if I'm wrong. Okay, but yeah, this ada
Maybe
Yes, right.
Yeah.
last year. Betul?
Yes, last year.
Tahun lepas Doktor kalau tak silap
Yeah.
kan?
Yes.
Okay. Alright, so nama dia Dr. Haniza kan? Alright, so I think all of you already know, I think we share the same identity. Okay, kita serumpun, we share the same identity, we have the same diversity of human, the people, right, in both country. Okay, so this is just to have a brief introduction for the university. So the University Pendidikan Sultan Idris is in Tanjung Malim. All right, so Tanjung Malim is a district. All right, so that fall in the state of Perak. All right, so Perak, and it is maybe around 90 kilometer or 100 kilometer from Kuala Lumpur. All right, so if you want to travel to Kuala Lumpur, it is quite easy in a way that we just use expressway. So it will take just maybe 1 hour or something to reach to Kuala Lumpur. Okay, all right. So okay. So before we go further into what are or what we can do with this microbial enzyme and things like that, all right, I would like to introduce to all of you, I think in fact all of you already know about this, okay, about the central dogma of life, all right, or central dogma of protein synthesis in this case. Okay, so when we talk about central dogma, everything has to be started from a material, right, that keeps information. And we do know that this material tak lain dan tak bukan, it is just the DNA, right, the nucleic acid, the DNA or the RNA. Okay, so in this central dogma, it started with the DNA. Okay, so with the DNA, then it will be transcribed, right? So transcribed to become the RNA. Okay, in specific, we call as mRNA, the messenger RNA. And this messenger RNA, with the help of a ribosome and things like that, all right, it will be translated to a form of protein. Okay, and this, even though this flow, it seems to be very, very general, right? Sangat umum. But when we refer to each particular protein, it is very, very specific, because like what I mentioned to all of you, all right, DNA is information keeper, all right, penyimpan maklumat genetik. Okay, and try to imagine in our body alone, all right, just to have an example, right, how many kinds of protein that is being synthesized every day, all right, 7 days a week, 24 hours a day, without non-stop. Because when this protein synthesis stops, or in other words, we call as the cell metabolism stops, right? We need to say that that is the time when kita mati lah, we die, right? And then everything stops. Okay, so as long as we live, all right, so this process will just happen and happen and happen all over again, cycle and cycle and cycle. Okay, and of course, even though each of the DNA part that we call as the gene, all right, in this case the gene, all right, will keep the information of one kind of protein, but we have many genes in our chromosome. Okay, and all of these genes, even though they produce, they translate into very, very specific kind of proteins. All right, the synthesized protein will actually they will communicate with each other. All right, so far I have not reached to one conclusion that one protein will just work by itself. Okay, all proteins will need to communicate, and they work together to make a cell live. Right, to make the metabolism works in a very, very what we call as a very dynamic way. All right, it is dynamic. Okay, so let's start at the first stage. Sekarang adalah transcription. All right, so in transcription, we as the scientists, right, will actually use different kind of stages, be it in transcription, translation, or even at the stage of regulation. We will try to make modification of it so that we can control the gene expression. Please allow me to use to draw a little bit. Okay, to actually create some of the traits or some of the characteristics that we want. All right, in this case, we want to change the gene expression. Okay, whether we want to have a lower expression or higher expression. Okay, we want to change the traits or characteristic of the protein that we want. All right, and of course, last but not least is, of course, we dedicate to the cell function. We want to change the cell function. All right, from A to B, for example. So all these things we can do through techniques, especially that involve molecular biology techniques. All right. Okay. So in this case, when we talk about molecular biology techniques, I'm sorry. Okay. So basically, we will work in many kind of stages, right? We work at the transcription level, the translation level, and to regulation level. Okay. So this genetic engineering work, all right, basically in my case, all right, I mean, it also depends on the person who work on that particular project. But in my case, I would always like to use the approach that we start from the transcription level, because at this stage I can play around with the main source of the genetic keeper. All right, that is the DNA. Okay, right. Okay, so in this case, what I do is that normally I modify the DNA. So what is meant by modification of DNA, right? So modification of DNA means mutagenesis. I mutate, all right? So we can do whether mutation, we have two types in general, all right? One is site-specific, one is random mutation, all right? So for site-specific, we have site-directed mutagenesis, okay? For the random, we have genome library, all right? We have the shotgun technique, all right, that we can actually, by chance, all right, to change some of the location in the DNA, in the gene in specific, all right, so that we could have the characteristic or the traits or the desire that we want, all right. So, but in my case, I would like to always use site-directed mutagenesis. The reason is because in site-directed mutagenesis, I can target, I go back to the first slide, right? I can actually target the site that I want, all right? Which site that I want. So in this case, you can see that this big structure over here, which is the beta 4 subunit, all right? And this is the interacting molecule, all right? So of course, when I want to mutate anything, I have to target at this site, because this interacting molecule actually affecting the function of the whole protein. Okay, I want it to have a better affinity. All right, for example, all right, saya nak dia punya interaction. All right, mula-mula asalnya mungkin maybe the interaction could be very weak. All right, secara originalnya. But when I already obtained this structure, I want it to become stronger. Okay, so in this case, I mutate the site where this IM interacts with the beta 4 so that they can have a better affinity, stronger interaction. Okay, so in this case, so that we want, right? But sometimes not all the strong interaction will give the desire that we want, the result that we want. All right, sometimes we want to make it loose, all right, supaya dia akan ada kita punya the result or the hypothesis that we wish for, basically. Okay, so it all depends on the mutagenesis. All right. Okay. All right. So next, all right, we can also regulate the transcription level. So when we regulate the transcription level, meaning to say that we can control the amount of RNA polymerase that being expressed, okay, because RNA polymerase is the one that could synthesize the mRNA at this stage. Okay, so in this case, in specific, it is mRNA. Okay, so while in the translation level, okay, we can do a post-transcriptional control or translational regulation as well. So what is meant by translational regulation in this slide is basically to have to control the amount of ribosome, okay, and also to the amount of certain protein, all right, that actually involve in the translation of mRNA to the protein, okay. And of course, when the protein, when the protein is finally synthesized, okay, we have the protein. We don't do anything at the transcription and the translation stages, but we do modification in terms of the protein, for the protein. And in this case, we normally use chemical modification. Okay, all right. And then finally, it comes to the protein, like what I mentioned, the characteristic 1, 2, 3 that we want. All right. So when we already investigate all these gene expression patterns, the traits, meaning to say that the phylogenetic of the synthesized protein, all right, we do check on that as well, because we want to relate the protein that is synthesized to their ancestor. Then we can see what are the actually the history that behind the synthesized behind the synthesized protein, and we can actually use the other protein to combine them together and make it as something like a family tree or something. All right. And of course, when we have these traits or patterns, all right, and the phylogenetic tree, all right, then we can actually predict the cell function or the protein function that maybe that work mostly to unknown proteins, all right, protein yang tidak pernah diketahui fungsinya, for example. Okay, so all these three characteristics we can combine to form basically one model that we call as interactome model. All right, later on I will tell you what is interactome model, because this interactome model is my, so I will say that this is my latest project, all right, in combining the information that I obtained from the DNA to the information that I obtained from the protein, and I combine them together to work as one. I would like to say I would like to build a model in a form of a pathway. What is this protein relate to other protein, and so on. Okay, so yeah, this is so far my focus in my laboratory right now. Okay, all right. So okay, so this is the overview of my presentation for today, the sharing for today, that is actually divided into 3 parts. Hopefully I can use this 1 hour, 1 to 2 hours to you know to share all the things over here, because yeah, I just afraid that it's going to be too dry if I just use, you know, like just giving talking alone. So that is why I always prefer, if possible, you can just interject anytime, not only to questions, but if you would like to give opinion on that, then probably we can have a good discussion, right, throughout the session. Okay, just please feel free. All right, please don't be shy. All right. Okay, so this process stages in this presentation are actually protein expression, all right, protein purification, and also lipase inhibitor. This lipase inhibitor is very a little bit different, all right, because sometimes I feel bored with what I do. I'm so sorry, a little bit bored, because when you know when you do a little bit more on your field of study, you do molecular biology, you work with a microorganism, you work with DNA, you know, like okay, so sometimes I want to do something that is a little bit different. Okay, that I work with the people from the chemistry department, all right, in UPSI, all right. So we develop inhibitor, all right, for an enzyme, all right, and this enzyme is not related to any microorganism. Okay, lipase, all right. So we got this enzyme, this lipase, we just simply bought it, all right, and we come up with we, not to say we create, we isolate and we characterize one of these inhibitor. Later on, I will just let you know what is it. To me, it's a little bit, personally, I feel it very interesting to me. Okay, because this lipase inhibitor is able, and it has the potential, right, to become the drug for anti-obesity treatment. Okay, and I would like to know why why obesity is one of our great enemy. All right, a very, very I would say a rival to us in Malaysia. Okay, just in Malaysia. All right. Okay, so before we start, okay, like what I promised to all of you, okay, I would actually like to do a quiz. It's just a very brief one. Don't worry. Okay, just a very, very brief one. Hold on, yeah. Want to keep patient. Okay, hold on, yeah. Okay, so I would like to have macam just to refresh our mind. All right, on the very, very basic knowledge about enzyme and also protein. Okay, so to join this, basically, semua dah masuk, ada yang masuk. Okay, I would like to I will just copy the link, all right, and paste it in the chat box so that you can just directly join the quiz using this link. All right, so I will paste it right now in the chat box. Okay, hold on, yeah. Okay. Okay, so I hope all of you already can see this in your chat box, the link. All right, and please feel free to join us now. All right, because yeah, throughout the quizzes, then probably we can discuss. All right, to refresh our mind. All right, so I hope this can be done very quickly, please. All right, so right now we have two, Yudha and also Rahma. So probably I hope more. Right, Opik, okay, Dr. Bowo, thank you so much. Okay. All right. Thank you so much for joining us. There are 6 participants right now. Okay, all right. Okay. Amida, okay, Dr. Umi, thank you so much. Okay, Sri, we have Ayu, we have Nur Hanifah, Will.
Silahkan yang lain ikut semua.
Yeah, please
Tinggal klik
feel free
di
to join us. We have around 95 participants, 96 for today. Right? So the thing is that, okay, all right, there is no correct or wrong answer in this, all right? So, I mean, just in case sometimes we do get wrong answers, right? But it's okay, because we do learn from the wrong things that we do, right? Okay, so proses belajar adalah kita buat kesilapan, then it's okay. So we can always discuss, all right? So that, this is not knowledge. So yeah, I hope we can help each other in this. Okay, so thank you so much. We have 29. Right, increasing right now. All right, Dita, we have Enda, all right, choose Nunia, all right, Shafiq, Rizky, okay, Ahmad Fakri, okay, Rama, thank you so much, Rida, okay, Fajria, Winda, thank you, Adha, Arbah, okay, so probably we will have another 1 minute, all right, because before we will start, all right, so we will start at 11:50. I mean, at your time is 10:52 a.m. Indonesian time. Okay, we have Danny. Thank you so much. B, Irbah. Okay, come on. Titi, thank you. All right. Gustina, Nana, thank you so much. Okay, Dr. Slamet, thank you so much for joining us. Fitria. Okay, Putri. Miss Shania, are you with us? So probably you can join us too.
Ramadan Microbiology.
Okay, thank you. All right, so when we reach 50, then we can start. Okay, Rima, thank you. Rizky, okay, Nikan, Putri, okay, thank you. All right, Sukarti, thank you so much. Okay, 48 right now, we have 2 more. Come on. Okay.
Ayah, adik-adik nanti dapat
Okay,
hadiah.
Shafan, thank you so much. Nafi. All right, okay. So probably you can start now. All right, but you can always join us. I mean, yeah, throughout the weekend. All right. Okay, so we will start now. Okay. So let's go to the first question. All right. Okay. Oh, hold on, yeah. Okay, are all proteins enzyme? So is it yes or no? True or false? Okay, so okay, 35, 36, 51. Okay, 36 participants already answered. So okay, we have finished this 20 seconds of it. So congratulations, all right, to all the one who actually answered. Okay, so we go to the next question. All right. Later on, we will check whether what is the answer. All right. So the next question. So ready? Yeah. Okay, 3, 2, 1. All right. Okay. All enzymes are protein. Dia soalan dia terbalik je. Okay. Whether it is true or false. Okay. All right. We have 6 seconds. All right. 3, 2, 1. Okay. All right. So next we can go to the next question. Hold on ya. Macam mana nak buat? Okay. Okay. All right. So then we go to the next question. All right. So for this question, 37 answered true and 8 answered false. Okay, so the next question is, okay, which one is not true? Which one is not true? Whether enzymes speed up chemical reaction, okay? Enzyme control one type of reaction or several types of reaction, right? Enzyme react best at the specific temperature. Enzymes are made of fatty acid. So the one that is being asked over here is false. All right, which one is false, basically? Okay. All right. Yeah. Okay, masa dah habis. All right. And then, all right. Try to do some application. Okay. All right. So we go to the next question. All right. Okay. There are 10 of it. Ada 10 soalan saja. All right. Okay. So what is another name for enzyme? Right? Whether it is a chemical catalyst, a microorganism, inorganic catalyst, or biological catalyst. So which one is the best that fit another name for enzymes? Okay, 3 seconds, 2 seconds. Okay. All right. Okay, time's up. Okay, so 42 of you. All right. Hold on, yeah. Okay, so right now the first in the chart, Risky. Okay, congratulations. But never mind, we have another berapa soalan lagi ya? We have another 6 questions. All right, so the next one is number 5. Okay. All right. So how do enzymes speed up chemical reaction? Whether increasing activation energy, increasing deactivation energy, decreasing activation energy, or decreasing deactivation energy. Right? Which one? How do they work? How do enzymes work? Right? To speed up chemical reaction. All right. Okay. Okay, time's up. All right. So we'll go to the chart. Siapa dulu? Okay, Risky still in the top, on the top. Right. Next is Will. Right. Congratulations. Okay, so we go to the soalan nombor 6. Okay, number 6. All right. What is a substance called if it is speed up a chemical reaction? Whether it is a reusable, a specific, a catalyst, or it is called as a fragile. Mana satu? Okay. All right. Okay. 44, 45, 46. All right. Okay. So let's look at the chart right now. Okay, Risky, Will, Shafan. Okay, Dr. Bowo. Okay, thank you so much. All right. Okay, catalyst. All right. Okay. So number 7. Okay, any substance that is called acted upon by an enzyme is called an. All right. What is the substance that act upon enzyme? Coenzyme, substrate, polypeptide, or vitamin? Okay. All right. 44. Okay, all right. Okay, all right. So we go to the next question. Okay. Okay, soalan nombor 8. Okay, we have 2 more to go. Okay, which one is not true? Which one is false? All right, enzyme can be denatured. All right, when the temperature gets too high, enzymes speed up chemical reaction. Enzymes can only be used once in a chemical reaction, and extreme pH can denature enzyme. So which one is false? Yang salah. Which statement is false? Okay, all right. So time's up. Then we can see right now. Okay, Will, congratulations. All right, on top right now. Okay, so 22 answered the third option. All right. Okay, number 9. Question number 9. Okay. Any enzyme speed up chemical reaction in a cell, but can only be used once. True or false? Okay, so this statement, whether they ask an enzyme speed up chemical reaction, but can only be used once. So whether it is true or false. Okay, all right. So let's see. Okay, all right. Okay. All right, so false, 33 answer false. All right, 14 answered true. Okay, so the last question, I believe. Okay, all right. Each enzyme has only one substrate that fit into the active site. So what it is called? All right, so whether it is called specificity, reusability, denaturation, or fragility. All right, so this is a statement. Each enzyme can only have one substrate in the active site. All right, so what word can best describe this characteristic? Okay, specificity, reusability, denaturation, and fragility. Okay, all right. Okay, so Will, all right, congratulations. All right, so 45 answered specificity. All right. Okay, so this is the last question. Do you want to end the quiz? So yes, okay. All right, so let's look at the score. Nothing appeared. Oh, okay. Okay, congratulations to. Hold on, yeah. I think my internet line is a little bit slow. Oh, okay. Congratulations to Will. All right, Tanya, Alhamdulillah. All right. So we have Will at the first place, Rizky, second place, third place, Shafan. Okay. All right. So thank you so much for the participation. And again, this is not to actually evaluate what we know about enzymes and things like that. Okay. So I would like to emphasize that this is just a refreshment session, all right, so that we can look into what we can understand and what I can let you know later on. All right. Okay, so let's look at the question. Kita tengok pada soalan sekejap. All right. Okay. We look at to review the question. All right. All right. So all proteins are enzymes. Okay. So basically the answer is true or false. That's it.
False.
False, all right? So not all proteins are enzymes because some proteins will have different function, okay? Some proteins may act as an antibody, right? Some protein may act as hormone, all right? And some they have, they work as structural molecule, for example, in our muscle, all right? So they are not enzymes. So the answer is false, basically, all right? Okay. So the next thing is all enzymes are protein. True or false?
False.
Some of you may say false. Some of you may say true. Right? Basically, this is still a debate. Right? I don't have, from my point of view, basically I don't say this as true or false because some of us, it depends on which fraction that we are. The scientists, right? Scientists pun ada fraction tau. Dia ada mangsab dia, right? Because some of the scientists will say that, yeah, all enzymes are proteins. But in the recent, not to say recent, 10 years ago, this research is about 10 years ago. Some of the scientists, they investigate on some of the RNA, all right, ribonucleic acid, and they exhibit this RNA, some of these RNA that they investigate exhibit the characteristic just like what enzymes do, all right? They catalyze reaction. They slow down the activation energy. So that is why some of us may agree, some of us may disagree. So in this case, we don't have orang kata apa, the basic conclusion, all right, the ultimate conclusion whether to say that whether all enzymes are protein or not, right? Because some of them may say some of the RNA are enzymes as well. Okay, all right. Okay, so next, which one is not true? All right, basically, of course, we can see that enzymes, all right, are made of fatty acids. So this one has not been proven so far. All right, so of course, among these 4 options, yang ni salah lah. All right, because fatty acid they make triacylglyceride. Okay, all right. Okay, next, what is another name for enzyme? So of course, it is biological catalyst, because some of the catalysts, they work not in biological system. They work in inorganic system, because some of the metal ions, for example, all right, they have this property of catalyst. So in this case, we call as chemical catalyst. All right. So if enzyme, because it is a biomolecule, so it is the best fit into as a biological catalyst. All right. So jawapan dia biological catalyst. Okay. How do enzymes speed up chemical reaction? All right, you speed up chemical reaction through decreasing activation energy. So about in any of the chemical reaction, they have certain activation energy. The higher the activation energy, the more difficult a product can be produced. All right. So in order to speed up the chemical reaction, kita mesti turunkan. We have to lower the activation energy so that the process become faster and more rapid. Right. In this case, so the answer is decreasing activation energy. Okay, so next, what is a substance that is called if it speed up chemical reaction? Of course, it is a catalyst, the more, I mean, the best, right? That fit into this option, kita panggil sebagai catalyst in general. Okay, all right. Any substance that is acted upon by an enzyme is called an substrate, right? So anything that actually act on the active site, kita panggil dia sebagai substrate. Right? Which one is not true? All right, basically, enzyme can be denatured when the temperature get higher. Yes, because enzyme is especially those which derive from protein, they will become denatured, just like the egg white. All right, bila kita masak, when we cook the egg white, then it become from colorless to white color, and they clump together. So this is actually the characteristic of the protein being denatured. All right, so enzyme speed up chemical reaction. Yes, of course, it's very, very true, right? Enzyme can only be used once in a chemical reaction. So this one is false, because enzyme are biomolecules that actually can be recycled over and over again. Okay, all right. And the last but not least, extreme pH can denature enzyme. Yes, right, because that is why in our body, for example, some of the enzyme will have different optimal pH. Right, for example, those enzymes that work in our stomach, they require a very, very low pH, very, very acidic pH, because in our stomach we have hydrochloric acid. Okay, some of the examples, for example, amylase, right, that is available in our saliva. Our saliva pH is around maybe a little bit alkaline, right, a little bit basic, right. So it only can be worked in a alkaline pH, all right. So if it goes into the stomach, then it will become denatured. Okay, the amylase will be denatured. All right. Okay, so the next question is, an enzyme speed up chemical reaction, but can only be used once, right? True or false? So the answer, of course, is false, because it can be recycled. Okay, again. All right, so each enzyme has only one substrate and it fit into the active site. So what it is called? It's called specificity. So it has to be specific, right? It's just the one that we normally call lock and key model, but basically lock and key model is no longer applicable nowadays, because we know that enzyme, especially that is made of protein, they are quite flexible in the structure. All right, they can change the active site according to the interaction that is formed between the enzymes and also the substrate. Right, so I would rather say that it's more to flexibility. All right, if we have an option over here is flexibility, then to me personally, I would say that it is more to flexibility. All right. Okay. Okay. So is this the last one? Oh yeah, this is the last one. Okay. So I hope with this, the brief quizzes, brief quiz, then we can have better understanding later on when I try to share something on the presentation later on. But of course, please again ask me questions. All right. Do you guys want to have a rest or something like that? Is it okay to continue?
Yes, okay to continue, Doctor.
Boleh okay.
Yeah.
All right. Thank you so much, Dr. Umi. Okay, so right now I would like to go to the first stage of the sharing session, which is on the protein expression. So this is a very early work in my project. You know, when we just entered the university, and we don't have any financial aid at the beginning. All right, so we will work on something that is very simple. All right. We start with a very simple experiment. Okay, in this case, I work with the protein expression. I check on certain protein on what is the level of expression, and then once I already know the expression, I set my own goal, whether what is the expression that I wanted to. All right, that is actually can be used in the industry. Okay, because when we talk about industry, they don't want to have a very low expression, because low expression, the issue is that they don't have enough material or the enzyme to be commercialized later on, all right, to be used in the bioreactor later on, right? So that is why I always work with recombinant enzyme. I isolate any gene that I want, all right. I do cloning and I put it into any of the microorganism, mostly bacteria, right, to produce this recombinant enzyme. So this recombinant enzyme can be produced in a very large scale, all right, dalam quantity yang lebih banyak, all right, compared to the wild type, all right, kita refer to the wild type. Okay, so as an introduction, okay, all of us, okay, I believe all of you over here, I mean, on the floor today, basically know what is the function of enzyme. Okay, in this case, when we talk about industry, all right, there are more sectors, right? But what is being focused in nowadays in the world is that we have these 5 sectors, all right? That is the pharmaceutical, that is normally used for the healthcare aspect, all right? Food processing, in which it is more into food security aspect, all right? Detergent, this detergent normally we use in a daily washing, all right? Biofuel, all right, biofuel comes to the very important discussion, because we know that petroleum, all right, that we use as a petrol, all right, as a source of energy, will actually exhaust, will actually finish up maybe in the future, all right. So we need to have alternative so that this petroleum can be replaced one day. Okay, and last but not least, paper and pulp. All right, this paper and pulp nowadays maybe not so much into focus, because most of our system right now actually looking into a more environmental friendly that we are going to use. I mean, right now, even in the Malaysian government, we seldom use a paper. All of it are actually online. All right, we just use soft copies, and in which I really support on this, because when we have this physical paper, all right, so there are a lot more trees that need to be cut. All right, so that's very dangerous for our biodiversity and also to the environment. Okay, so in this case, when we focus on these 5 sectors, all right, we have many types of enzyme that we can use. All right, for example, we can use lipase, protease, all right, trypsin, all right, cellulase, xylanases, amylases, all right. So if you if we really look into this category of enzyme that I give over here, all right, yeah, ada kat sini, all right. Basically, most of these enzymes, they are hydrolases, all right, meaning to say that they break down a large molecule into smaller molecule, but water must be present, all right. Air mesti ada, sebab kalau tak ada air, if there is no water molecule present in the reaction, meaning to say that the reaction will not work. So that is why enzymes like proteases, lipases, amylases. What else? Papain. All right, papain is part of protease. All right, amylases, cellulase and xylanase. All right, they are all actually falls under the group of hydrolases. Okay, and of course, when each of these enzymes they work in this particular sector, so this would be the application in general. Okay, for example, when we talk about food processing, the juices that we drink, for example, right, basically some of the juices, especially the fruit juice can, all right, most of them, they are derived from starch, all right. So when starch, because the end user would be the human, all right, so we need to have certain taste of it. Kita perlu rasakan makanan or minuman, the food or the drink that we consume, all right, to become tastier, all right, untuk ini apa kepuasan diri, all right. So all this, when we for example drink fruit juices, we want it to be sweeter, all right. So that is why all this enzyme comes into play, all right, that actually convert the starch to glucose. So this is hydrolase, all right, to change sucrose, for example, into fructose corn syrup, all right, and to produce some of the prebiotic and probiotic food supplement for human. Okay, for example, okay, detergent. All right, so this is one of my very fond focus, right, in my research as well, because most of us, when we use detergent, all right, be it to wash dishes, all right, be it to wash our clothes or something like that, right, little of us may know, some of the brand in detergent will actually integrate enzymes, right, into their detergents, right, because these enzymes, right, for example, proteases and also lipases, they act to remove stain, they act to remove fat and oil. For example, lipase, all right, lipase is actually will have a very specific in this function, all right. Protease, on the other hand, will actually be used in to remove some protein stain in our clothes, for example, blood, for example, all right. So this is very important as well. Of course, not just the enzyme alone. Enzyme will just acting as the accessory in the cleaning punya process. But of course, the main cleaning process will be done by the detergent lah. For example, SDS ke, for example, SLS ke, for example, all right, sodium lauryl sulfate or sodium daldecyl sulfate. All right, so all these detergent, they have an amphipathic property that will dissolve in water and also dissolve in hydrophobic condition. So that is why the process, the cleaning process is most done by the detergent, the SLS or the SDS. Okay. All right. So the next thing is basically, okay. Let's look at the global market. All right, why enzymes is being focused in the world nowadays. All right, so from the publication that I searched, okay, in the recent one is 2018. I believe there are more recent one, but this one I found it in 2018. All right, if you look in this global enzyme market, so most of it, when I see these numbers, all right, they have basically the same they are expanding in a similar volume. All right. For example, if you look into food, animal feed, detergent, and also some of the technical enzymes, for example, the one that involved in pharmaceutical, detergent, and paper and pulp. So all these are technical enzymes. They are expanding in a similar pattern. All right. From 2016, that is the actual year where they obtained this data, and the projection in year 2021, which was last year lah. All right. So this is a projection around maybe 300 millions or 400 millions. From 2016 to 2021, 5 years projection. Okay, and again, when I talk, because my focus is mostly on detergent protein, all right, so I look into this technical market. All right, if we translate into this data over here, all right, of course, if you look in North America, they are still being the champion basically. All right, nowadays, all right, 2021. But yeah, we as in the Asia Pacific, all right, it's not losing as well, right? Because we have a very rich biodiversity. we can actually isolate more enzymes from the microorganism or from the plant or from the animal, and this can be actually cloned and put it into a bacteria so that it can be produced more and more and more and more. All right. So that is why we are expanding as well in Asia Pacific, including Malaysia and also Indonesia. All right. So the one that you can see over here, the EMEA means in Europe countries, Middle East country, and also Africa. So all these three continents, they combine together. So that is why it looks very, very high over here, all right, because there are three continents. But if we separate it into individual continents, so basically the number can become less lah. All right. So I would say personally, if this EMEA is not combined, so Asia Pacific could be the second main player, all right, for the technical enzyme in the world, all right. So in this case, okay. So that is why it is technical enzyme is one of my main focus, because it has multiple functions, all right, in our daily life and also in the industry as well. Okay. All right. So this is what that has been mentioned by Ibu Dr. Umi, all right, that mentioned that basically the very first enzymes, all right, actually derived from animal, all right, the rennin, all right. So the rennin over here. So for enzyme, all right, in this case for the sharing today, I focus on proteases, right? So proteases is the enzyme that falls under the enzyme commission of 3.4.221 to 26. So ini adalah kategori enzyme. So enzyme they do have identification, all right, macam our own identification lah, right? So this identification is based on the function that they have, all right? So basically 3 means they falls into hydrolase, 4 means what kind of reaction that they catalyze. For example, protease, they catalyze the breaking down of a peptide bond, all right? So it falls number 4, right? So 21 to 26 means they have different characteristics during the catalysis. For example, whether they are metalloprotease, they are acid protease, alkaline protease, and so on, all right? So they catalyze hydrolysis of protein and also peptide, the proteases, right? And it is obligatory, dia mesti ada dalam semua organism, because if they absent in the organism, so protein that we consume, for example, will not be hydrolyzed, and also it cannot form energy to us, right? Meaning to say that the metabolic reaction of protein will not happen. And if this is not happening, so actually we cannot live, we will die, right? Sebab kita tak ada source of energy, right? We cannot just depend on carbohydrate. We cannot just depend on fatty acid as a source of energy. Remember, because if you answered the quiz just now, one of the question asking, are all enzymes protein, or all protein enzymes, all right? Because enzymes or protein, all right, they work more than as an enzyme, right? They work as hormone, they work as antibody, they work as a structural component in our body, all right? So they must be added, okay? So last but not least, when I relate this EC, all right, is actually, this part 21 to 26, actually is actually referring to this one, all right? Whether they are metalloprotease, serine protease, cysteine protease, or aspartic acid proteases, okay? So then we look into the sources, all right? Dia ada, these proteases, dia ada dalam animal, plant, and also of course in microorganism, the microbial proteases. But why nowadays we actually emphasize on microbial enzymes, or in particular microbial proteases? Because microorganism, they have this very special characteristic that animal and plant cell do not possess. For example, bacteria, right? They can propagate very fast. In 24 hours, they can propagate into millions or trillions of cells, right? However, for animal, they can't do that. Even for plant, they can't do that, right? So they have a very slow growth rate, right? So in order to have to harvest enough enzymes for industrial application, so basically we need to wait longer time, all right? So in industry, time is very precious, sangat-sangat berharga. So they don't have enough time to wait for this animal and plants to produce enough enzymes. All right. So, and of course, when we deal with higher organism, they need to have a bigger space in this case. All right. So the animal, if they want to have a cattle, I mean a group of cattle and things like that, and in fact for plant, they need to have a bigger land, so that they can feed into this animal and to breed the plant or the animal. But for microorganism, we can just culture it in a tank, a very small tank basically. All right. But of course, when we go into the industry, we talk about bioreactor that actually feed in 100 liter, 1000 liter, and so on. All right. So there is a bigger one. So when you have a bigger space, bigger tank, meaning to say that you can culture more and more of the microorganism, in specific bacteria. All right. So what would be the application of enzymes? Application of protease, all right, in specific. So it can be used in a peptide synthesis. So peptide synthesis is one of the product, all right, that is used in pharmaceutical, especially in the cosmeceutical industry. Cosmeceuticals means bidang kecantikan. So we normally take it as a supplement for health supplement, and most of it is dedicated to our skin health. All right. So peptide is very important, especially the collagen, right? The collagen kita makan tu. All right. Of course, the detergent, because one of the application is to remove the protein stain, for example, the blood stain. All right. In dairy and also bakery. All right. Especially in the production of cheese, like what Dr. Umi mentioned, branding, for example, is used in cheese. All right. And of course, in leather. Because proteases, one of the example is keratinases, right? Keratin, right? Because kita punya leather, animal leather lah. In our case, the human skin, all right, possess keratin, all right? Keratin is the one, or elastin, all right? So this elastin or keratin is the one that actually give structure to our skin, all right? Some of us over the time, all right, we get old, all right, and we have a very, very loose skin, right? Yang lebih longgar kan? Because the reason is because our cell produce less and less elastin and also keratin. Okay, meaning to say that sometimes more proteases is being synthesized in the cell to break down this keratin and also elastin. So our skin become less elastic in the case when we get older. All right. So in this case, for the leather industry, of course, proteases is used untuk melembutkan, to soften the texture of the leather for bags, for example. We don't want our bag to be very, very stiff, right? All right. So kita perlukan lembut lah. So in this case, the protease will act to break down certain elastin or keratin in the animal skin to make it softer. All right. Okay. So far, is there any question on this? I mean, from the early slide up to this one. Ada soalan?
Silahkan teman-teman yang ada pertanyaan bisa disampaikan ke Dr. Akil.
Silakan, betul. Silakan. Ada soalan?
Sepertinya belum ada, Dr.
Belum
Akil.
ada,
Mungkin
okay.
nanti di bagian sesi diskusi.
Okay, alright. Okay, thank you so much, Dr. Umi. Alright, so please allow me to continue lah kalau macam tu Alright. Okay, alright. So now we are going a little bit detail, right, about the work that I do in my lab, basically. Okay, so just now I introduced you on protease. So I believe you guys already know that. Okay, basically I work mostly in proteases. All right. So where does this protease in my work comes from? Dari mana asal usulnya this protease? All right. So the protease that I'm working right now, okay, is actually derived from a bacterium, right? That is called Pseudomonas aeruginosa. Okay. So this is also a local isolate. That has been isolated by one of the senior in the lab, right? Previously isolated, right, in Selangor, Malaysia, and this Pseudomonas is a little bit different because it produces a very, very nice colony, right? It produces water soluble and also green fluorescent pigment. Because if I put this under the UV light, dia akan jadi fluorescent green, very nice fluorescent green. All right, so this is the colony of Pseudomonas aeruginosa, and we call it in our lab we call it as Pseudomonas aeruginosa strain K. All right, so this is, of course, this is a Pseudomonas. They are gram-negative bacterium. All right, so people may ask, well, okay, this is just a Pseudomonas. because Pseudomonas is very ubiquitous. It is abundant. Dia sangat-sangat banyak, and it actually can be easily isolated sebenarnya. Alright, in the soil, basically. It is abundant in the soil. But what is so special about this Pseudomonas? The speciality of this Pseudomonas compared to other Pseudomonas that has been actually reported so far is this Pseudomonas is able to grow in organic solvent, right? In particular, the solvent that we grow this bacterium is in the BTEX. All right, BTEX is benzene, toluene, ethyl benzene. Let me write it. All right, benzene, toluene. All right, ethyl benzene. Ethyl benzene, and also xylene. Okay, and for your information, these 4 organic solvent, they are polyaromatic hydrocarbon, right? PAH, polyaromatic hydrocarbon. Then this polycyclic aromatic hydrocarbon, they are normally the compound that is found in the water as a water pollutant. Dia sebagai pollutant dalam air. Alright. So when this microorganism is able to live in this BTEX, right? So it has the characteristic of being degrading, right? Of degrading this PAH, meaning to say that it can be used in bioremediation processes. Okay, itu pada bacteria. But to me, even though I graduate from as a microbiologist, I don't actually have so much fond interest into looking into the bacteria itself. Okay, I like to look into what kind of product that is produced by this microorganism, not just Pseudomonas, but any kind of microorganism. All right. In particular, I look into protein because my area of expert is in protein chemistry. Okay. All right. Then what we did is that when we culture this microorganism into the BTEX, all right, it grew. All right. So when it grew, it is the proof that it can degrade benzene, toluene, ethyl benzene, and also xylene. Right? So we need to say that there are certain enzymes that is being produced by this Pseudomonas to degrade all these 4 compounds. Right? When degradation of 4 compounds happen, meaning to say that benzene ini ataupun toluene ataupun ethyl benzene or the xylene can become less toxic to the environment. Sebab benda tu dah diuraikan, is being broken down. Right? So it become less toxic into the environment. So what we did is that we try to isolate the protein, okay, or in specific the proteases from these Pseudomonas. All right. So eventually we found that through 2 processes of purification method. All right, we isolated 3 types of protein. All right, P1, P2, and P3 referring to the protein. All right. And when we have these 3 proteins, all right, we again run the function, all right, of this protein in organic solvent, basically isooctane, decane, dodecane. And I would like to inform all of you that basically enzymes, tak kisah protease, lipase, or any kind of enzyme, normally they denature in organic solvent. All right, kenapa? Nanti saya akan cerita. I will let you know why actually protein can denature in organic solvent. Right, and the focus of looking into organic solvent tolerant enzymes or proteases is very important in the industry, because some of the industry, they use a very high amount of organic solvent. Right, if we use the enzyme that cannot tolerate in organic solvent, it will be useless as well. Sebab enzyme akan mati, and of course organic solvent are the one that actually kills the enzyme. Right, so that is the reason why the journey or the discovery of enzymes, proteases, for example, that can be tolerated in organic solvent is very important. Okay. All right. So in this case, we try to look into 3 of these enzymes. All right, and we run the activity. All right. So by enzyme is, if you remember, it is a biocatalyst. It has activity. All right. So we run it in different kind of 3 types of organic solvent. Basically isooctane, decane, and also dodecane. And it seems that P1 and P3 are the one that possess high level of activity in organic solvent, meaning to say that P1 and P3, dia ada potensi, it has high potential to be used in industry yang memerlukan organic solvent, right? For example, peptide synthesis, right? So of course, if you look over here, P1 and P3, between P1 and P3, P3 is better, okay, because it possesses higher activity compared to P1. However, we try to investigate this P3. So far, it is so unfortunate to let all of you know that this has not been successful so far. It cannot be expressed so far lah. Okay, so that is why we change our focus to P1, right? Even though it has less, but it worth to be investigated further. All right. So next, what we are going to do, we did is that we want to see what is P1 basically, right? Because it has proteolytic activity, maksudnya dia adalah protease, all right? But we don't know what is the name of this protease, all right? So from here, all right, okay, so before that, I would like to let you know why enzymes in organic solvent is very, very important. Okay, so this figure basically showing one enzymes, all right? This enzyme is a protease that we call as trypsin, all right? So this trypsin, because trypsin is hydrolase, all right? If you remember what I mentioned to you just now, hydrolases requires water molecule. Dia perlukan air, all right? So at the first panel over here on your left, all right, this trypsin, all right, is soaked into water, all right? So the blue circle over here, yang bola yang berbentuk biru ni, all right, they are actually water molecule. All right, so this water molecule basically in the normal condition, they will surround the surface of the enzyme or protein. Why this water molecule is very important on surrounding the protein is due to hydration. Protein perlukan air, dia perlukan hydrate. Macam manusia, we need to be hydrated. Right? Same goes for enzymes. They need to be hydrated as well. Okay. So basically this is just a simulation, the simulation. All right. So but when this trypsin is subjected to organic solvent, for example, in this case is octanol. THF is tetrahydrofuran, and ACN is acetonitrile. So all these three are actually organic solvent. Okay, so what is the difference between octanol, tetrahydrofuran, and also acetonitrile? Okay, so the difference is that how well this organic solvent can dissolve in water, and it is actually measured by a unit that we call as log PO over W. It is the measurement of a partition coefficient of a solvent, for example, in octanol, all right, in octanol water two-phase system, okay, meaning to say that the more negative the value of the log POW, meaning to say that it can dissolve in water easily, right? So that is why it says that over here, meaning to say, if you compare this OCT, THF, and ACN together, ACN will have a more negative log POW, meaning to say that ACN will dissolve in water more easily than THF and also ACT. And if you see over here, all right, basically, can anybody, I mean, before I explain, can anybody have a general overview or a guess, a wild guess? What is the effect of OCT, THF, and ACN in these three figures? If you want to compare to the in water, can you guys guess basically? Just briefly, just a comparison in this figure, these four figures. Anybody can get a wild guess? Can take a wild guess, right? Apa kesan dia? What is the kesan? What is the effect of ACN on the enzymes? What is the effect of THF for the enzyme? What is the effect of OCT for the enzymes? Anybody can take a wild guess? Ada? Shania, I can see your face. So can you take a wild guess? Just a wild guess. It's okay to be wrong. I mean, it's okay. Apa effect dia? Apa kesan dia of OCT, THF, and also ACN? By the colors. Yang merah ni adalah OCT. Merah is OCT, this merah is THF, and this one is ACN. And of course, the blue one will still be water lah.
Correct me if I'm wrong.
Yeah, it's okay.
The blue side decreased by the red side.
Yes. Very good, Shania. Very good. Well done. Because Shania mentioned to us, yes, the region of red color, all right, become more and more when we go to OCT, THF, and ACN. You see, the region of this ACN is more compared to the THF and also OCT. Why? Because if, like what I mentioned just now, ACN has more negative log POW, meaning to say that the toxicity of more negative log POW, right, will create more and more detrimental effect on the enzyme. Okay, meaning to say that the toxicity will increase when the log POW is getting more negative. All right, so ACN is the most negative over here. So if you see this simulation, basically the ACN molecule are actually stripping the water molecule on the surface of the enzyme. Okay, you see, inactivation due to stripping of essential water from the enzymes. So on the surface of this enzyme, we call as essential water molecule. All right, so these ACN are actually stripping the water. All right, so once water molecule is being stripped on the surface of the enzyme, meaning to say the enzyme will be denatured. Right, dia akan denature. Denaturation means it no longer work, right? All right, so this is what happened. All right, so that is why my goal in the lab is to look for, or if possible, to modify the enzymes so that it can be stable in a very negative log POW organic solvent, right? Because most of the industry, they will use water soluble organic solvent, for example, methanol, right? And methanol, for your information, it has the most negative, one of the most negative organic solvent, okay, in the chart of log POW. All right, so that is very important. Okay, so next we look into the enzyme that we isolate for this P1. All right, so for your information, we are talking about P1 right now because P3, even though it has more stable activity in the organic solvent, but we fail to express this protein. All right. Okay, then we work into the purification of P1 protein. All right, so P1 protein, eventually, if you want to know what is this protein is all about, basically we did an experiment that we call as amino acid sequencing, because sequencing is not just for DNA and also RNA. We can do sequencing for protein as well, but it's going to be very expensive. All right, so in this case, I just managed to sequence, all right, 9 amino acid, and it cost me quite an amount as well. Okay, and eventually from this amino acid, 9 amino acid, all right, so basically this is the size of the protein after purification. Basically 2 stages of purification, dual stage, all right, ultrafiltration and also HIC. HIC is hydrophobic interaction chromatography. All right, and once I got this sequence of the amino acid, I blast it. All right, eventually it gives me the enzymes that we call as elastase. All right, remember when I talk about elastase, it is the protease that degrade elastin. All right, and of course this elastase derived from Pseudomonas. All right, okay. So the next thing that I do is that basically to fish out the gene, to isolate the gene. Okay, so this gene is isolated because it can be easily isolated because it is well-known enzymes, well-known protein, right? But again, the special characteristic is that it is able to be stable in organic solvent. All right, so I fish out the gene, right? So basically, this gene is 33 kilodalton, and basically, I think the size of this gene is around, hold on, is around 500 amino acid, right? Including starting from the signal peptide, pro sequence and mature sequence, and also the stacking region. Stacking region is a region whereby it is a stop codon is there. So stop codon is over here. All right. So signal peptide, why signal peptide is appeared in this protease is due to the reason is because it is hydrolase. All right. Hydrolase normally are the enzymes that secreted out of the cell. They are extracellular enzymes, and this signal peptide is very, very important because it will actually recognize the protein that is being synthesized in the cell to be secreted out of the cell. All right. Dia akan hantar this enzyme, it will deliver this enzyme to the membrane of the cell and eventually be secreted out. All right. So pro sequence is the sequence that help in the folding of the enzyme into the functional form. And of course, the mature sequence, just 301 amino acids, around 30 to 33 kilodalton. All right. This is the mature enzyme. So this is the sequence that actually codes for the active enzyme. All right. So this gene, right, actually clone into a vector. All right. This vector, I think most of you, I mean, those who actually go into the lab and work in molecular biology, they are very, very, very well versed with a vector. There is called as PUC19, PUC19. But in this case, I don't use PUC19. I use PUCP19. So this PUCP19 is very, very important and very, very special because this vector is called as a shuttle vector. All right. Shuttle vector is a kind of vector that can actually propagate and express recombinant enzyme in more than one host, right? Because normally we clone it in E. coli, all right? But in our case, in my case right now, I don't want it to be cloned in E. coli, all right? I want it to clone in a host that more than E. coli, all right? I want to clone it back into Pseudomonas. I want to clone it back into different hosts, for example, Klebsiella or something like that, all right? Because we do have the reason why, if you remember the P3 protein, we have the problem, the issue of the recombinant protein cannot be expressed in E. coli. All right, and some of the literature mentioned to us that those enzymes or those proteins that actually derive from Pseudomonas, they're very, very difficult to be to be inserted or expressed in E. coli because they have different codon usage, they have different expression system, right, in Pseudomonas. So that is why I choose to use PUCP19, a shuttle vector that can be actually propagate and express protein in E. coli and also in Pseudomonas. All right. Okay, so this is the early investigation of what I did. All right, to look into what is the expression system of this recombinant enzyme. All right, I clone it into E. coli. All right, in specific, I clone it into top 10, a strain. All right, and a strain that we get called as Cara-X. All right, so both of these strains can be purchased commercially. Boleh beli je. All right, okay. And for Pseudomonas, I clone it into PO1. PO1 is a very, very well-known Pseudomonas strain, whereby the full genome sequence of Pseudomonas is obtained from this particular strain. All right, that I bought it from ATCC. ATCC is, I think most of you know about this as well. This is a culture collection center. All right, American type culture collection that we can just purchase this particular bacterium. All right, and also S5. All right, S5 cannot be purchased. All right, it is just available in my lab. All right, because this S5 Pseudomonas, it is also a BTEX tolerant bacteria. All right, so this bacteria can be used in bioremediation as well. So that is why I use this S5. All right, so from the early stage of the expression, we can see the fold over here compared to the control. All right, so for E. coli, the highest is just 13-fold, around 14-fold. And for Pseudomonas, it's just 5-fold. All right. To me, maybe this is not really enough. All right. I aim higher. I aim to have at least 15-fold or more, or 20-fold, right, compared to the control. All right. So what I did is that in order to, from my hypothesis, what I did is that I'm thinking whether, whether I should change the expression system. Whether I should change this vector. All right. I want to add in a better promoter. I want to add in a better tag, for example, hoping that I can have higher expression level. It means to say that I want to have overexpression of the protein. Okay. So what I did is that I changed the promoter. All right. I changed the promoter. At this early stage, the promoter that is used in this PUCP19 is PLAC, right? PLAC is P-lactose, promoter lactose, and this is a very leaky promoter, right? Leaky promoter means that even without any induction, all right? In this case, I put the inducer as IPTG, all right? So even in the absence of inducer, the protein can still be expressed, right? Because leaky expression sometimes could cause issue in terms of the energy of the cell, right? Because we want to preserve the energy of the host, E. coli, for example, because we want to dedicate all the energy to be just used for the expression of the protease, elastase, all right? So in this case, I wanted to make it tighter, right? So I changed the promoter into the promoter that I call as a T7, right? T7A1O4O3 promoter. So this is a very, very tight promoter, all right? Meaning to say that without the presence of the inducer, the IPTG, the gene or the protein cannot be synthesized or expressed. Okay, so why is it tight? Because it possesses two operator systems. Okay, operator, if you look into this lag operon system, if you look into lag operon system, they are actually the switch to turn on or to turn off. If I only have one operator, meaning to say that I can easily turn on and off the system. If I have two switch, right, ada dua switch kan, right, meaning to say that I have more control, right? I can control, I can tightly repress the expression of the protein. So ada dua operator, right? So this is what I got, all right? Okay, so it is a little bit confusing. I'm so sorry, but this is actually the one that I construct, right? So I actually, again, I started with PUCP19, all right? But I integrate another vector that I got from one of the collaborators. He is very, very generous enough to give this vector to me for free, the PSS213, all right? Because this PSS213 possess this promoter, right? The T7A1O4O3 promoter, the type promoter repressor system. All right. Okay. Then I come up with basically, I think I come up with five series of vector system. All right. The first one is Picon 2. All right. The second one is Picon 230. Hold on, yeah. Saya pun lupa. Okay, this one. Okay, the third one is Picon 23 kanamycin, Picon 4, and also Picon 5. All right. So all of these five series, five different kind of promoter, they have different characteristics. All right. If I mention to you all, it's going to be very, very lengthy. All right. But in general, all right, these five vectors, all right, they have different characteristics. For example, this one, this characteristic, they can be grown. It can be cultured in the presence of an antibiotic we call as zeosin. Zeosin tak ramai orang pakai in bacteria, but most of them they can use it in yeast system. All right, for example, Saccharomyces cerevisiae. All right, so normally they use zeosin as the selecting marker. All right, for the recombinant form of the yeast. All right, so in this case, I use this particular vector to be cloned into the yeast system. All right. However, for kanamycin, all right, kanamycin is another antibiotic, so this can be used as a additional selection marker in bacteria. And Picon 4 and Picon 5, they have different characteristics. All right. So yeah, so these are the five vectors that I constructed. Okay, and these all five vectors has been patented, and the patent was approved 2 years ago. All right. And still enforced. So alhamdulillah, so there are five vectors of this, all right, but that has been patented. Okay, all right. So what I did is that I checked on all these five series of the expression vector, right? Again, all right, in E. coli and also in Pseudomonas, all right. So what I have over here, all right, basically, remember that my target is around 15 to 24 increase. All right, but this time after the modification of the vector or the vector that we constructed in the lab, all right, basically the highest that we can get over here is around 82-fold, all right, much higher than what I expect. Alhamdulillah. All right. Whereby for Pseudomonas, I have around 15-fold. So yeah, we are trying to have this more and more. We are working on this basically on the Pseudomonas especially. All right. So later on, yeah, we have so-called enough level of expressed protein at the bench level, at the lab level. Okay. So what we did is that we go on with the purification method. All right. We construct. We try to design what kind of purification, the best purification that we can use in order to have the highest purification level, the purity level, and also to decrease the level of losses. Because when we have more and more stages of purification, of course, we will experience high purity, but at the same time, we will lose protein throughout the process of the purification, basically. All right. Eventually, we optimize the purification method, and there are two. We finalize two stages only. All right, which is the HIC, the hydrophobic interaction chromatography, as the first step, and then next we go into the second step, that is the IEX. IEX is ion exchange chromatography. All right. Okay. So what we can see over here after ion exchange chromatography is that we managed to get this purity of enzyme around more than 99% of purity, all right, based on what we did on the SDS page, both non-denaturing and also denaturing, and also the native page, all right. Polyacrylamide gel electrophoresis, we can see just a single band over here, all right. So we need to say that, of course, maybe if we have load more and more protein, we can see tiny band, but basically we can see what we can see over here is 99, more than 99%, all right, of purity. Okay. So then this is the ultimate experiment, right? Whether we can see the purified protein is suitable to be used in the industry, whereby we carried out characterization experiment. Okay, so this characterization basically will work on the physical and also the chemical characterization, because we need to fit into the parameter that normally be used in the industry, right, for the enzymes, right? So in terms of the thermal stability, right? So basically what I can say is that for this recombinant, because I compared the recombinant and the wild type, all right? So basically the thermal stability is getting lesser, right, in the recombinant, probably because during the expression of the protein itself, there might be certain changes in the structure of the cell, in the structure of the promoter, structure of the protein, right, whereby this does not affect on the activity, but it does affect on the thermal stability, all right. However, what is being investigated, my focus in this characterization, of course, thermal stability, optimal pH, pH stability, meta-ion, and so on is very, very important. But my focus basically will be just on this one, all right, organic solvent stability, all right. I am looking for the enzyme that can be stable in hydrophilic organic solvent, meaning to say that organic solvent that can be easily dissolved in water. For example, DMSO, methanol, and also ethanol, right? Methanol, ethanol can be senang je kan nak dissolve this organic solvent in water, all right? But if you refer to my early slide that we work on the simulation, okay, this water-soluble organic solvent will have a very, very negative log POW, and they are very, very toxic to the cell, to the protein, okay. So right now, I have the protein, the recombinant one, right, that is very, very stable in organic solvent that is normally can kill the enzyme, right, which is these three organic solvent, okay. Yeah, this is a characteristic. Well, it is a good news, right, but this is not enough to really support why, because we as the academician, we are not actually satisfied with just this result alone, right? We need to investigate why and how basically the organic solvent, the protein can be stable in this organic solvent, all right? And we don't run simulation, because simulation is basically, at the early stage, we can run simulation, but right now it is not the level of simulation anymore, but we move into the wet lab, all right, that we investigate why this protein, the protease, the elastase, can be stable in hydrophilic organic solvent, okay. So the next thing that I did was I ran a circular dichroism, analysis. Circular dichroism analysis is the analysis that actually work on to look into how the organic solvent change the secondary structure of the protein, because we know that protein is consisted of 4 structure: the primary structure, the secondary structure, tertiary structure, and also the quaternary. But in my case, the elastase just reach until the tertiary structure. All right. Okay. So I work in various concentration of methanol, 0%, 25%, 50%, 75%, and also 90%. All right, the highest one. So I can see when the changes from 50 to 75% of methanol, there's a very, very large changes in terms of the activity. All right, from 0 to 50, the activity is still there. All right, the enzyme is still work in up to 50% of methanol. Tapi bila saya increase, when I increase the methanol to 75%, then there is a sudden drop of the activity. So then I look into why this happened, right? Through the circular dichroism, right? So from here, we can see that when I increase the concentration of methanol, basically the alpha helix, the secondary structure. So secondary structure ada dua kan, the main one, all right? Alpha and also beta. Okay, so I can see that the alpha helical structure drop when I increase the concentration of methanol. So once this alpha helix drop, it become unordered. Unordered means random. All right, of course lah, sebab dia change kan, they change the structure, it become unordered. Unordered ni dia tak ada function. All right, so it give me the indication that this organic solvent it plays around just with the alpha helical structure, because I can see that the beta structure and the third structure are stabilized even when I increase up to 90%. All right. So what happened over here is the alpha helical structure. Okay. All right. So this is just to check. Sorry. This is another protein. All right. That I work with amino acylase. All right. This is another protein that is amino acylase. So this amino acylase is a kind of protease as well. But this protease work at the terminal part of the protein, the amino part. Sebab dalam amino acid kan kita ada carboxylic part and also the amino part, right? So this enzyme work at the terminal amino part. All right. This is actually a thermophilic bacteria. All right. Basically, it can be stable up to around 70 degrees centigrade. Right. My master student worked on this one. All right. It was like maybe 3 years ago work on this. All right. That we isolated this enzyme and basically we purified this enzyme. All right, we purified this enzyme and it shows that it has a higher stability at the 70% of the temperature. All right, so this is a thermophilic enzymes. All right, okay. So next, we move back to the elastase. All right, okay. So this elastase, all right. Right now I already know. Okay, the methanol will affect on the secondary structure, which is the alpha helical structure of the enzyme. All right. So now I want to check on the structure. Okay. I want to know how this elastase look like in 3D. All right. Remember during my at my first slide. All right. Some of you may doubt whether I did move the slide or not. Okay. I'm so sorry for that. Basically, the structure that we can see is actually the crystal structure. So these images that you can see over here, yang ada kat sini, right, di sini dan sini, right, actually they are protein crystal. Okay, we try on different technique on protein crystallization technique. Dia ada banyak, all right. So we just try 3, all right. We use crystal screening, we use hanging drop, and we use counter. Okay, hold on, yeah. Okay, we use another method. This method has not been used by most of the crystallographer. All right, that is called capillary counter diffusion. This method is in collaboration with our Japanese collaborator, JAXA, Japanese Aerospace Exploration Agency, because we try to bring this protein to be crystallized in ISS, in a microgravity environment, the angkasa lepas. Okay, we bring this protein to be crystallized, and in fact, the image that you can see over here is actually the image that is crystallized in ISS, International Space Station. Inilah dia, all right. And when we diffract this crystal, dia daripada angkasa lepas, dia turun, okay, and then we diffract, and then eventually we got this the structure, all right, that is solved at 1.39 angstrom. Okay, so this is quite fairly good resolution to be solved and to get the 3D structure. Okay, why, some of you may ask, okay, crystallization can be done on the ground, in the earth, on earth, but why do we need to bring to the outer space? The reason is because we want to check on what is the effect of microgravity towards the packing of the crystal structure. Because when the crystal structure getting packed, meaning to say that the molecule of the protein is getting closer and closer together, we can have higher resolution. All right, but if we have a protein that is packed loosely, far away from each another, okay, so we will have a very, very bad resolution. Meaning to say that sometimes when we get the result of it, it could be doubtful whether is this the real structure or just basically a rough structure of it. Okay, right? So that is the reason why we conduct the experiment in a microgravity environment. All right. Okay. So yeah, I think what is the time right now? Okay. So
the times is about 12. But we only have 30 minutes left. Maybe
okay.
we move to discussion or yeah.
Okay.
Yeah.
Okay, Dr. Umi.
Yeah.
So thank you so much.
Yes. The next maybe for the purification and also for the
lipase.
Yeah. Dr. Akil can share it for the next session in the next weeks. It's okay.
Okay, but I think for this second and third part is going to be not as much as the first part.
Oh yeah.
And I think I can end it in within half an hour. Is that possible?
Yeah, it's okay. If Dr. Akil not need time to take a break, it's okay.
I mean, yeah, if you want to have a break, then it's okay.
Now, for Dr. Akil, if you want to take a drink or maybe to have take a break for a while, yeah, it's okay. Because yeah, so you can have a drink.
Yeah, because I'm so sorry. Probably, yeah. When I talk, sometimes it's very difficult to stop.
Yes, it's not no problem, doctor, because we can also get more information about the protein engineering. Okay, maybe yeah. If there any sufficient time, maybe Dr. Akin can continue the explanation. But if there some student want to ask question, maybe is there any student want to ask a question before we continue the lecture?
Yes, please.
You can raise hand.
Probably I will skip this purification stage because doctor, I think this purification stage I can do it at the second round because I planned for the second round of our lecture next week, inshallah next week. I will try to explain more into how protein is purified and how protein can be immobilized, because immobilization is part of the very, very important stages in protein in constructing a protein to be used for industry.
Oh yeah, it's no problem, doctor. So maybe it will be more comprehensive if we can get the material for the next weeks.
Sure, yeah. Inshallah, yes.
Okay, maybe we now go to the discussion session. Is there anyone who want to ask or sharing something from their experiment or their research in protein and want to ask to Dr. Akil?
Or maybe I suggest if I can finish the last session, which
is
then we can go to the discussion.
Yeah,
around 10 minutes or 15 minutes at most.
Yeah, it's no problem, doctor. You can go on to continue the topic. Okay.
All right.
Yeah,
okay,
thank you. Yeah, thank you so much. Yeah,
okay, thank you. All right. So yeah, I will skip this one for next week. All right, because this is to me I'm so sorry. I think this is quite important to me as well. I mean, this is my interest because I work with one of the tech for purification, and it actually did it patented because this is one of the very important findings. All right, in one of my projects. Okay, never mind. We skip this one and we go to the last part. All right, which is lipase inhibitor. Remember that I got bored with what I did throughout the years. All right, I mean, near to 15 to 20 years of working with molecular biology, working with proteases and things like that. All right. Okay, so I try to change a little bit on my focus into inhibitors. All right, inhibitors are actually the compound because the name is inhibitor. So it is actually compound that actually stop the function of any enzymes, right? Inhibitors, they inhibit, all right? So in my case, I focus on anti-obesity because in particular, the enzyme that we use, all right? I mean, in our body, right? Lipase is an enzyme that break down fat into glycerol and also fatty acid, right? Of course, this fatty acid will be absorbed in our body, and it can be used whether as a source of energy or as the deposit, right? Deposit means that it can become the layer of fat in our body, all right? And when this level of fat could not be controlled, meaning to say that we did not control our diet and we consume every kind of food that we take in, right? So basically, the lipase will work to degrade the fat into fatty acid because we don't move that much, and this fat will be deposited. The fatty acid will be deposited back as fat in our body. All right. So and when this become uncontrollable. All right. So it become obese or fat. All right, in our body. All right. Okay. So this is the thing that I'm quite actually embarrassed to actually share, but this is fact, right? And we are in science, so we have to share this. This is one of the statistics that are obtained in one of the newspaper article that is published in Malaysia, all right, by saying that the obesity is the epidemic in Malaysia, all right, whereby we can see that in this chart, right, in Malaysia, the obese people actually constituted half, all right, compared to the normal weight or the underweight community, all right, half of it, right, half of it. Okay, so it's actually 1-to-1 lah in this case, overweight and obesity against normal and underweight is 1-to-1. All right. So if you look at in this figure, basically Malaysia is over here, and it's quite embarrassing to say that we have a very high, right? We have a very high rate of overweight and also obese people in Malaysia. Congratulations to Indonesia, because your color seems to be lighter, much lighter compared to Malaysia. Okay, so you guys are doing very good job. Congratulations, Indonesian. All right, so yeah, please don't follow Malaysia because Malaysia probably just love to eat so much, I think. But if we compare to other countries in the Western country, for example, in this case, the North America, all right, they are quite they are the highest basically, right? Okay. But if I bring this data back into Malaysia alone, all right, around 73% of the death are due to NCD. NCD is non-communicable disease. For example, like a cardiovascular disease, diabetes, cancer, chronic respiratory diseases, and all these NCDs, major NCDs, are actually contributed by the body weight, all right, whether it is an overweight or obese patient. So most of the obese, I would say, they may be contracted with this NCD, one of these NCD, okay? And sad to say that most of the younger generation nowadays, all right, are the major contributor towards the overweight and obese part, all right, the statistics, all right, in Malaysia, all right, 30 tahun, all right, and up to 70 years old, but as young as 30 years old, right? And I think right now we have the rate of less than 30 years old of getting obese, all right? So, all right, sorry. All right. So the risk of being obese or overweight, of course, I think most of us know about this, is actually the diabetes, especially the type 2 diabetes, right? High blood pressure, okay, cancer, gallbladder disease, and also fatty liver disease. Okay, so nowadays what people do to actually overcome this obesity is basically by just to change our lifestyle, right? To look into our diet, what we should eat. All right, so all these are basically based on our psychology, right? How are we going to train our brain, the central nervous system, to actually control our desire to eat more and more and more, right? So another way to do it is by surgical approach, right? Whereby there are certain surgical, for example, liposuction. For example, to some of the celebrities, especially, they now opted for to shorten the length of their small intestine, hoping to say that to reduce the amount of fatty acid that's being absorbed, all right, from their small intestine. Okay, and last but not least, through drugs, right, that we call as pharmacotherapy approaches, all right, they're actually targeted towards the central nervous system, right, the adipose tissue, and also the gastrointestinal tract, right. Okay, so far the most being used is the pharmacotherapy through the drug, right. Okay, so drug, for example, like the one that we can see over here, okay, they have been tested in clinical stages, but they fail. All of this fail. All right. So some of this generic name of the drug, they target on central nervous system. They target on stomach. Right. So most of it they target on central nervous system. Right. Except one drug that is the only one available. Right. In the market. Right. Is Orlistat. Orlistat is a drug that that only acted in gastrointestinal tract, whereby this drug, commercial drug, is acting as inhibitor for lipase, right? The one that I'm looking for right now. Okay, and most of these drugs, why they fail in the clinical trial is due to all these reason, right? People die, right? People have side effects, right? After consuming this drug, right? In the clinical, right? Even though some of the drug are being sold in a black market, right, underground market, right. So this those drug that is not approved, so it's very important, very dangerous, right, and it causes a very serious warning to the consumer, to the human, for example. Okay, so in this case, I look into alternative for the lipase inhibitor, whereby I look into one of the plant that we call as agarwood. In Bahasa Melayu, is we call as gaharu. All right, gaharu is very well known in some of the Middle East country, that they use it is as a burning material to have a very fragrant smell. All right. Okay. So this gaharu, right? Basically, we obtain the compound. Right. Just a very simple one explanation. Right. And eventually, we managed to isolate 4 compounds. Right. C1, C2, C3, and C4. And these 4 compounds are mostly they are flavonoid, all right. And we actually did some experiment to inhibit the lipase, and this lipase is obtained from porcine. Porcine means swine, all right, because most of the experiment when we use the lipase is actually derived from the swine, all right, because they have a little bit similarity in terms of the function compared to the human lipase, all right, and it shows a very, very good inhibition, whereby if you look into this graph, right, we can see that compound 1, 2, and also 4, they actually managed to suppress the activity of this porcine lipase, right, until less than 10%. So they are very, very strong inhibitor. And when we ran the simulation, and it shows a very, very strong interaction of the compound. So this is the compound that we use, all right. So this is compound C1, all right. We dock the compound C1 with the structure of this porcine lipase, all right, and basically it shows a very, very strong interaction, meaning to say that yes, whether it is in a wet lab or in the simulation, it shows a very relevant correlated observation between the simulation and also in the wet lab, right? So this, we are trying to commercialize this yeah, we try to actually modify some of this structure over here, right? For better docking, for better interaction with the porcine lipase. All right. So yeah, I think I've for today I end this session with by providing an acknowledgement and special thanks to my collaborator in UNY. All right, Jogja. All right, to JAXA, the Japanese Aerospace Exploration Agency, for bringing the protein to be crystallized in the space station. All right, Kyoto University, University Putra Malaysia, and of course to my affiliation. All right, University Pendidikan Sultan Idris, and also to my counterpart in the Malaysian Genome Institute. All right. So with that, I thank all of you for the time today to listen and to listen to my sharing. Okay, and I pass it back the mic back to Dr. Umi. Please, Dr. Umi.
Thank you very much, Dr. Akil, for the nice presentation and very interesting topic about the enzyme. And now I would like to invite the audience who want to ask a question. Boleh dalam bahasa Indonesia, silahkan adik-adik atau teman-teman mahasiswa yang ingin bertanya, atau mungkin bisa dalam bahasa Inggris. Silahkan, masih ada kesempatan untuk bertanya, kami tunggu. Karena topiknya sangat menarik ini tadi ya, dokter.
Terima kasih, dokter. Silakan. If you wish to have like if you don't to use the mic, probably you can just put it in the chat box. It's okay.
Yeah.
Ada chat box pun okay.
Maybe the topic about the genetic engineering is not familiar for us in education. Our students, have to learn about the pedagogy and also in biology. But I think it's interesting, the latest information in enzyme, the latest research in enzyme provided by Dr. Akil. Oke, silahkan teman-teman. Silahkan. Oh ya, ini sepertinya ada. Ya, ada. Ya, one question from Mbak Rian Hanatan. Okay. She want to ask about what is the product that Pseudomonas aeruginosa can produce in our environment. Okay, thank you. I think, yeah. Okay.
All right.
Maybe,
thank you, Dr. Umi. So probably, thank you, Dr. Umi, and also thank you to Riani. Okay.
Rianhe.
Thank you so much, Rianhe. All right. Thank you so much. All right. So for the question, it's very important question. All right. So Pseudomonas is actually, like what I mentioned, is a very ubiquitous banyak. All right. It's easily found in the soil. All right. And they act in the environment as actually most of it as a degrader. All right, degrader in terms of pollutant degrader. Okay, and they are very important. That is the reason why they can exist in most of the area. Okay, because they help us by cleaning the environment. That is why I choose this Pseudomonas, because it can produce many kind of enzymes, many kinds of protein that can actually help to degrade most of the pollutant that is available in our surrounding. Okay. In fact, if you look into some of the publication, sesetengah terbitan, all right, they use this Pseudomonas as one of the consortium, satu kumpulan of bacteria yang digunakan untuk dia membersihkan, contohnya macam oil spill bahan minyak. So one of the bacteria. Okay. So there are many products, many protein that is produced by this Pseudomonas, especially dedicated to clean the environment. It's okay.
Oke, begitu Mbak Rian.
Okay. Right. Did I answer your question, Rian? All right. Okay. You're welcome. Most welcome.
Mungkin yang lain teman-teman silahkan jika ada pertanyaan untuk Dr. Akil.
Boleh izin bertanya, dan Dr.
Iya,
Akil.
silahkan Ibu. Iya, silahkan Ibu.
Silakan.
Silahkan
Sebelumnya,
Ibu.
perkenalkan, nama saya Baiti Nur Hanifah dari Magister Pendidikan Biologi UNS. Sini izin bertanya, tadi kan sebelumnya ada izin menyampaikan dalam bentuk bahasa Indonesia, nggih?
Boleh.
Ini tadi kan ada kuis yang menyatakan bahwa enzim itu tidak sepenuhnya dari protein, gitu. Atau mungkin sebagian protein atau memang seluruhnya itu bukan dari protein. Tetapi kan fungsinya dari enzim sendiri itu sebenarnya kan untuk mengikat atau mengikat protein, sehingga nah mungkin dari Dokter Akil bisa menjelaskan seperti apa gitu. Mungkin kan tadi Dokter Akil juga sudah menjelaskan bahwa itu masih kontroversial sebenarnya. Cuman yang masih diragukan itu dalam bentuk apa gitu.
Iya.
Betul, I
Okay.
hardly can hear what Ibu Betty asked. But thank you
Yeah.
so much, Ibu Betty. But for me,
Yeah.
your help to explain.
Yeah, I will try to help to say with Dr. Akil. Ya, Miss Betty bertanya, kenapa tidak semua enzim itu berupa protein, begitu? Apakah ada bentuk lain dari enzim selain protein, begitu?
Okay. Alright, okay. I got the question. Okay. Thank you so much again, Ibu Betty and also Dr. Umi. So basically, enzymes, right? Most of them they appeared in a protein form. Most of them, okay. If you remember, kalau Ibu Betty masa saya terangkan mengenai enzim itu, sesetengah saintis sebenarnya menemui enzim ini adalah sebahagiannya juga adalah RNA. RNA is ribonucleic acid. Di mana RNA ini dia mempunyai fungsi yang lebih kurang sama macam seperti enzim. Di mana RNA ini boleh mengubah substrate kepada produk dan juga merendahkan tenaga pengaktifan sesuatu reaction sesetengah RNA. Tapi secara umumnya enzim adalah terdiri daripada protein. Sebab itu kaedah untuk mendapatkan 3D structure, struktur 3 dimensi enzim itu sangat penting. Sebab kita daripada struktur 3 dimensi itu baru kita boleh lihat kepada bahagian mana ataupun area mana pada protein tersebut yang kita boleh olah ataupun modifikasikan untuk menjadikan sesuatu protein itu lebih bagus ataupun menjadikan dia kepada bentuk yang lain ataupun fungsi yang berbeza.
Ya, juga menarik itu dokter, kalau misalnya dalam bentuk RNA saja sudah bisa menjadi enzim begitu ya. Nah itu apakah juga ada sisi aktif, apakah juga ada bagian koenzimnya begitu kalau dalam bentuk RNA?
Okay, so soalan yang bagus Dr. Umi. Jadi untuk buat masa ini peranan ataupun fungsi RNA sebagai enzim cuma dari segi function sahaja, dari segi fungsi sahaja. Tapi dari segi mekanisme macam yang kita buat untuk docking, macam mana RNA boleh bertindak balas dengan sesuatu substrate, masih belum diketahui. Dia punya ilmu pengetahuan ataupun mekanismenya masih belum diketahui. Cuma dari fungsinya betul, ada fungsi yang boleh berperanan seperti enzim protein, enzim dalam protein.
Okay. Yeah, thank you very much, Dr. Akil. Okay, yeah, this is new information for us that RNA can be act as an enzyme. Yeah. Okay, is there any question? Ada pertanyaan lagi dari teman-teman? Silahkan. Ini menarik banget ini tadi. Okay, one question from me, Doctor.
Yes.
Yeah. Okay, in the genetic engineering, protein engineering, we see that the yield of the protein engineering at the beginning only 13-fold and also 5-fold in E. coli strain and also in Pseudomonas strain. So Dr. Akil tried to increase the number of the yield of the protein by choose the
changing the
choose
promoter system.
Promoter system, and also choose the vector.
Yeah.
Okay, choose the vector.
Yes.
So
Yes.
just in the promoter system, it should be at some substance which can repress or can induce the gene expression in the protein yielding.
Yeah. The reason why basically thank you so much, Dr. Umi, for the question. Basically, the reason why I throughout the investigation of this increasing of the expression fold, the yield of the synthesized recombinant protein is, I think, is probably is due to the energy that is kept in the host itself. Because at the early stage where I discovered that the fold is just around 13 fold, all right. Basically, because the promoter that is used is a leaky promoter. So when leaky promoter is there, most of the energy is used during the expression, even though there's no inducer. Because inducer is very important for the expression of the recombinant protein. All right, because if the promoter is really strong, if you don't add in any inducer, basically the protein will not be expressed. Okay. Meaning to say that if the protein is expressed, energy is being used. All right. So in a leaky condition, all right, no inducer, no inducer. All right, but expression still occurs. Meaning to say that the host, the cell, is still using the energy. A lot of them to synthesize the protein. But when I change the promoter to a very tight promoter, okay, so without any inducer, the protein will not be expressed, and meaning to say that most of the energy will be kept, and it will only be used when the inducer is there. All right, so that is why the energy is actually focused more during the presence of inducer, and the reason why it is, and that is one of the reasons why the expression fold is increased up to 82%, 82-fold. Compared to the control. It's one of the reasons is due to the energy that is kept in the cell.
In the cell, okay, yeah. Okay, thank you, Dr. Akil. Oke, ya masih ada lagi. Silakan Bapak Ibu teman-teman yang gabung di sini. Mungkin ada yang mau ditanyakan. Tadi topik terakhir itu menarik sekali ya. Ini lipase inhibitor gitu. Lipase inhibitor for treatment obesity and also diabetes mellitus. Yeah. So far, just in Malaysia or Indonesia, to treat this kind of disease using lipase inhibitor from tadi Dr. Akil itu from agarwood. Yeah. Is there any other source from medicinal herbal which also produce a lipase inhibitor in Malaysia maybe?
So far, of course, there are more herbs that is being investigated, but actually, when we talk about anti-obesity, not so many researchers are looking into the natural compound that actually targeted specifically at anti-obesity. Most of them they target on diabetes. They target on cardiovascular diseases. All right. So the reason why we don't target specific on this diabetes and also maybe cardiovascular, because that is not the main reason why it happened alone. All right. So that is why my team, together with the biochemistry department, we look into a bigger picture of it. What is actually the root, the source of this CNS happen? All right, the cardiovascular, why it happened, why diabetes mellitus happened. It actually rooted from one source. Is due to the weight, body weight, that actually what we call as obese or overweight lah, Dr. Umi.
Yeah.
So that is why we target directly into anti-obesity. So back to your question, whether is there any compound, natural compound that has been investigated by other researchers in terms of anti-obesity? So basically, there is no, because most of them, they target specifically on the disease. For example, diabetes. If you ask me on whether there is natural compound on anti-diabetes, yes, there's a lot of them working on it. All right, but if it is on anti-obesity, so I would say that so far through my knowledge, there is no publication yet. All right, that's target on specific compound on anti-obesity.
Okay.
Yeah.
So
This project alone, I
inhibit.
think we have managed to publish like around 5 publications.
Oh, yeah.
Alhamdulillah, we managed to do that.
Yeah. Because in Indonesia and also Malaysia have many different kinds of herbal medicine. So we need to explore their bioactive compound especially for treatment obesity.
Yes. Yeah. Actually, there are
Okay.
more. Yeah.
Ya. Oke, masih ada lagi. Silakan teman-teman, kesempatan terakhir untuk bertanya
Sila.
ke Dr. Akil.
Bu Umi, sekiranya saya ingin bertanya.
Baik, silahkan.
Bahasa Indonesia gitu.
Ya, semoga.
Nah, saya ingin bertanya nih, Bro. Kan ada tuh beberapa enzim yang diproduksi secara massal atau secara industri ya. Nah, misalnya itu produksi asam sitrat. Dalam produksi asam sitrat kan itu dengan cara menghambat enzim isosikratid dehydrogenase atau di dalam situs Scrap. Nah, itu dalam proses produksi massalnya itu bagaimana ya? Padahal kan itu terjadi pada tingkat sel. Ya, mungkin itu Bu Umi. Terima kasih.
Oh ya. Mr. Sifak asked about how to produce citric acid in cellular level to tadi untuk menghambat ini ya Mas, menghambat siklus Krebs atau gimana? Mas Sifak.
Kan ceritanya enzim kan ada yang diproduksi secara industri. Nah, misalnya kan enzim sitrat. Nah, enzim sitrat itu kan dari siklusnya kan pembuatannya dari dengan menghambat fermentasi itu. Nah, yang saya tanyakan itu bagaimana proses produksi secara massalnya. Padahal itu kan terjadi pada tingkat sel gitu loh, Bu.
Okay. He asked about how to produce in a large scale of the isocitrate enzyme while it produced in a cellular level, so can be produced in a large scale for the industry, maybe.
Okay. All right. So thank you so much, Shifa, for the question, and also Dr. Umi for the translation. Okay. So basically, not only actually dedicated to the particular product that Shifa referred just now. Tak semestinya. Alright. Sebab semua jenis enzim ataupun protein, alright. Sebab kenapa saya suka pakai protein? Of course kita punya fokus adalah enzim. Tapi bila kita bercerita mengenai industri, okay, semua jenis protein, alright, tak semestinya enzim, alright. Ini termasuklah hormon, termasuklah antibiotik, termasuklah antibody, alright, boleh dihasilkan secara industri dalam kuantiti yang sangat besar. So saya suka bagi penerangan dalam protein lah, sebab benda fokus ataupun subjek yang Syifa bagikan tadi sebenarnya dia boleh diguna pakai juga. Okay. Contohnya macam sekarang kita lihat apa yang saya buat di makmal saya adalah sebenarnya adalah di peringkat makmal yang kita panggil sebagai lab scale. Alright, lab scale ni dalam kuantiti yang kecil, dalam botol yang kecil lah, dalam paling banyak pun 1 liter ataupun paling banyak pun mungkin 10 liter. Alright. Tapi bila kita dah ada data-data yang kita pakai dalam yang kita ada, yang dihasilkan daripada lab scale, kita kena bawa dia untuk upscale. So, kita kena buat lagi satu eksperimen sebab dia sebenarnya berperingkat daripada lab scale pergi bioreactor. Bioreactor pun ada saiz dia. Alright. So, mungkin dalam 50 liter ke, mungkin ada 30 liter kan, yang lebih besar daripada lab scale. So, kita akan dapatkan data. Alright. Sama adakah data yang kita pakai untuk 30 liter tu berpadanan tak dengan yang 10 liter yang kita buat dalam lab. Right? Andai kata dia berpadanan maksudnya dia meningkat, hasilnya meningkat. Right? Barulah kita akan bawa ke level industri. Level industri ni saiz dia sangat-sangat besar. Dia boleh 100 liter, 1000 liter, lebih pun ada. So kalau kita pergi ke industri, kadang-kadang bila kita tengok bioreactor, bekas fermenternya tu, dia lebih tinggi, dia mungkin jadi 2 tingkat, tingginya macam 2 tingkat bangunan. So dia sangat tinggi dan sangat besar. Alright, so you can culture a lot of them, and once it is cultured, then benda tu of course perlukan purification. Alright, dan yang saya rasa saya boleh terangkan in the next coming lecture. So itu sangat penting. So mengenai dengan soalan yang Syifa tanya tadi, ia memang boleh dilakukan dan memang biasa dilakukan pun untuk pengeluaran yang lebih besar. Did I answer?
Ya, begitu Mas Syifa. Gimana? Mungkin ada feedback? Jadi memang harus ada optimasi. The first optimization first, and then setelah dapat yang optimal baru dibuat skala besar begitu.
Yes. Yeah. The term is optimized. Yes, it's correct.
Yeah. Gitu. Okay, thank you. One more question in the chat room, Dr. Akil. This is from our graduate student. Her name is Corisa. She want to ask about the protein breakdown by enzymatic process, especially proteolytic enzyme will cause change in the smell, texture, and appearance of meat or some fish. What causes such condition? Okay.
Thank you so much, Corisa. Another good question by students. All right. Thank you so much, Dr. Umi, as well. Okay, so yes, it's very, very true that when protein changes, all right, ataupun when protein denatured, it will create some smell, it will change the texture. The reason why is because it's due to the changes in the structure of the protein, in which, for example, when we use heat, for example, or even the enzymatic reaction alone, all right, it will create some bad smell sometimes, it will create some good smell sometimes, all right. It is due to the product after the breaking down. Depends on the type of amino acid or the peptide that is produced after the breakdown. Okay, different peptide, for example, they will give different texture. Different peptide, they will give different fragrance, for example. Okay, I would like to give the example macam the egg white, for example. Okay, the uncooked egg white seems to be colorless, right? It seems to be liquid in structure, right? But once we subject heat, kita letak api, kita bagi haba, alright, the egg white become solid and it changes the color to white, right? And it changes the smell as well. It's not as the smell is not as fishy as the uncooked one, right? Yeah. So the reason is because the peptide has been broken down. It changed to different thing. It changed to different peptide. It can be changed to a smaller length of peptide, or it can change to something from the protein form into the denatured protein form. All right. So this is the reason why some of the texture, the color, the fragrance can be different due to the different treatment. All right. It can be treatment by the protein, by the enzyme. It can be treatment by heat. It can be treatment by the pH. Macam-macam lagi. All right. And it's all due to the product that is being produced after each of the treatment. So about each of the treatment we have different characteristics.
Okay, thank you, Dr. Akil. Okay, that's all. Okay, I think it's a very interesting discussion about the enzyme and protein, but unfortunately our time has limited, and we will continue to the next topic in the next weeks with the same times ya, Dokter, jam pukul 10 jika di Indonesia, mungkin kalau di sana pukul 11 begitu.
Yes, inshallah.
Okay.
Or perhaps Dr. Umi, because just now I just had a meeting an ad hoc meeting. So that's why I asked, for it to be start at 11. So perhaps we can have an original time whereby we can start at 10 a.m. Malaysian time and 9 a.m. Indonesian time next week.
One hour more earlier. Okay, it's no problem, Doctor.
Yes.
Okay. Yeah. Thank you very much for today, for the interesting lecture today that give us more valuable insight and more valuable knowledge about the enzyme, and so I can conclude that from the Dr. Akil presentation that we can produce protein not only from the organism, native organism, but also we can produce protein from the other host organism by genetic engineering, by increasing their expression in molecular level. So, okay, ladies and gentlemen, thank you very much for your attention. And for Dr. Akil, once again, I appreciate, before we stop this session, I would like to show the certificate appreciation. Oke, Mas Wildan mohon dibantu dan sudah ada sertifikatnya.
Maybe next week's.
Oh yeah, yeah.
We have one.
Okay. One. Okay. I'm sorry, doctor. Maybe the certificate will be given for the next weeks. Okay. Okay. Thank you, Pak Bowa. Yeah. In this lecture, don't forget next week we will still have one more session with Dr. Akil. Start at 9 a.m. in Indonesia, and of course it will be more interesting about enzyme. And today I'm as a moderator. I thank you very much for your attention. I apologize if there are some mistakes in guiding this session. Thank you very much. We give it back to the Master of Ceremony. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the speech, Dr. Akil and Dr. Umi. Distinguished ladies and gentlemen, Alhamdulillah, we have now come to the end of visiting lecturer. On behalf of all the committee members and the college, I would like to express my gratitude to all the distinguished guests and participants for making this event successful. Before we part, please forgive us for our shortcomings. Till we meet again, perhaps in the next visiting lecturer second session will be held on Thursday, 7 April. Okay, thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much. Okay, thank you.
Yeah, thank you very much, Doctor. Yeah, see you next week. Yeah.
I'm sorry, Mr. Akil. Maybe
yes,
for the next week, you can inform to your student to join our lectures in the next session. Okay, thank you very much.
Sure. Yeah.
Okay.
Okay, thank you very much. Yeah, thank you. Yeah.
See you next week.
See you next week. Waalaikumsalam warahmatullahi wabarakatuh. Matur nuwun Pak Bowo. Berarti minggu depan.
Terima kasih semuanya, Bu Umi, teman-teman, Bapak Ibu yang lain. Ini tadi banyak sekali yang ikut ternyata.
Iya. Insya Allah minggu depan nanti saya sampaikan mungkin sampai jam 11 begitu Pak Bowo. Tidak. Karena beliau semangat sekali menyampaikan.
Sekaligus diingatkan mahasiswanya untuk bisa bergabung kita, Bu Umi.